Thank you for watching this video. My name is Sergio Doria-Belenguer and I will be talking about our last project, which is titled The Axis of Biology, a novel axis-based network embedding approach to decipher the fundamental mechanisms of the cell. Advances in capturing technologies has yielded to a vast amount of data. This data is often represented as a network in which nodes represents biological entities and where edges represents different type of interactions between these entities. Network models are a powerful approach to model different interactions that occur in a cell. For instance, protein-protein interaction networks represents the physical interaction between different proteins that give rise for the fundamental functions of the cell. While networks are a powerful source of information, we need to develop new algorithms in order to uncover the information that is hidden in their wiring patterns. However, in the last years, biological networks are becoming more complex and hard to decipher. Thus, a growing body of approaches using network embedding to simplify, visualize, and also facilitate the analysis of biological networks. The intuition behind network embeddings is to map nodes to a low-dimensional space where the geometry of the space reflects the similarity of the nodes in the original network. There exist a lot of different network embeddings approaches, but for this presentation I will be focusing on the non-negative matrix trifactorization, or NMTF for short. NMTF takes as an input a non-negative matrix and it approximates this matrix as the product of three low-dimensional matrix factors. P times S corresponds to the coordinates of the genes in the embedding space. And the matrix factor B corresponds to the axis that spans the embedding space where the genes are embedded. NMTF has several advantages over other deep neural network-based approaches. For instance, we can add an orthonormativity constraint to the basis, to the axis of the embedding space, and by doing that, we will reduce the dependencies of the axis. By reducing these dependencies, the organization of the entities in the embedding space is known to be better. But this is only one of the advantages. NMTF also allows for data integration. We can use different object data together. It is also considered as an AI explainable model. And finally, the non-negativity of the spaces generated by NMTF are known to better capture the functions that occur within a cell. After generating the embedding space and the representation of the genes in the embedding space, current embedding approaches mainly focus on the genes representation to develop different downstream tasks. For instance, one of the downstream tasks that are usually applied for is the capture of the cell's functional organization. Current embedding approaches, also called as gene-centric, captures the cell's functional organization by first clustering genes whose embedding vectors are close in the embedding space, and then by assessing the statistical overrepresentation of different cellular functions, usually represented as annotations like GOTOMs or reactome, across the gene clusters. Those functions that are overrepresented are then used to describe the functional organization of the cell. However, gene-centric approaches have several limitations for this task. For instance, the number of functions that are enriched across the gene clusters are known to be low. But not only that, it is also known that the redundancy of the functions that are enriched is high, meaning that the same function can be enriched in a lot of different gene clusters. Altogether, it reduces the functional information that we have for describing the cell's functional organization. But this is not the only problem. Gene-centric approaches, as I said before, relies on different ontologies, and because of that, they also carry with the limitations, with the intrinsic limitations that the ontology has. Current ontologies are defined by a predefined set of annotation, which means that you cannot discover new cellular functions that are not included in the ontologies but could arise from the data. The second problem that the ontologies has is that the connectivity of the terms in the ontologies reflects their semantic similarity, meaning that annotations that are connected if they are describing similar processes. But these connections does not imply a functional interaction between different processes, and because of that, functional interactions that give rise for higher processes cannot be uncovered. To overcome these limitations, in this work we propose to shift the exploration of the embedding space from the genes to the axis of the embedding space itself. To achieve this, we propose the following approach, an axis-based approach. The intuition behind our approach is to project genes functions to the space where both genes and functions are embedded. And we do it as follows. Let's have as an input a toy network X, and let's generate an embedding space, having in this case embedding space with the representation of the genes in it. As a first step, we propose to project functions to this embedding space, and we do it as follows. We fix the axis of the embedding space, so the most important thing of the space that spans this embedding space. Then we represent the functions of the genes by their Go terms, having a matrix of Go terms times genes. And then by doing this decomposition, we obtain the matrix factor U. This matrix factor U corresponds to the coordinates of the functions in the embedding space. Thus, after this step, we have genes and functions organized in the same embedding space. As a second step, we will project each of the functions to the axis of the embedding space, and then we assess for the statistical significance of these projections by the following permutation-based approach. We will generate a random representation of each of the functions in the embedding space and project this random representation to the axis of the space, repeating this process 100k times, having at the end a distribution of values for each function and each axis. And from this distribution, we can compute the p-value. We will consider for a function a projection to be statistically significant if the p-value is lower than 0.05. I have here to say something, and is that in our case, we only allow a function to be projected to one axis. This means that in the case that the p-value is lower for more than one axis, we will associate the function to the axis for which it has the highest embedding score. We apply this method for all the different functions in the embedding space, having at the end a list of axes with its corresponding associations. In our project, we apply this method to capture the functional organization of the cell from different protein-protein interaction networks. However, for this presentation, I will be focusing on the results for human. Also, in our paper, we also apply different embedding approaches. But in this case, I will be focusing on NTF since it's the approach that gives to us the best results. The first thing that we assess after applying our method to the PPI of human is to assess the ability that our method has to capture and uncover biological information from network embeddings. And we quantify that by the percentage of axes that has at least one associated Go term and the percentage of Go terms from the total that are associated with the axis. As you can see in this plot, almost 70% of the axes have at least one Go term associated, and almost 50% of the total Go terms are associated with the axis. This means that our method captures biological information from the embedding space. The next question was if the stratification of this information across the axis is biologically coherent, and we assess that by analyzing if Go terms associated with the same axis have higher semantic similarity than those associated with different axes. What we observed is here in the plot is that on average, Go terms that are in the same axis have 2.6 times higher semantic similarity than those that are associated with different axes. In conclusion, we saw that the axis of the embedding space uncovered biological information from network embeddings and that this information is coherently stratified across the axes. Having demonstrated that, our question was to investigate the biological meaning of the GO terms that are associated with the same axis. And before going inside the results, I would like to recall that interactions between GO terms in the ontologies reflects their functional similarity. In other words, their semantic similarity. Based on that, our first question is to understand if the functional interactions that our axes capture between the GO terms are in agreement with the ontology. And to do that, we can compute their semantic similarity. As you can see here in the plot, what we observe is that the vast majority of the GO terms associated with the same axis are functionally related and thus in agreement with ontology. But something that it was more interesting is that although this is true for the vast majority of the interactions, we also find some of them that are not included in the ontology, that arises from the data and are not included, as I said, in the ontology. Thus, our question is that these new interactions that we are capturing are meaningful. What do they mean? Are biological coherent? And we validate that by literature curation. More specifically, we validate 13 axes with the lowest semantic similarity among the associated GO terms. What we observe here is one example. This is an example of the axis 116 that has 4 GO terms associated. And as you can see here in a fast look is that the semantic similarity between the terms is low, meaning that they are not connected in the ontology. In fact, they are far away in the ontology. However, from a functional point of view, the first thing that we observe is that they can be grouped in 2 different small clusters. A big cluster that groups GO terms that participate in the regulation of the RNA transcription, and another one that is connected with the midbrain development. Interestingly, the functional interaction between these 2 groups is coherent since it has been demonstrated that the regulation of rRNA is important for the development of different issues in the brain. In conclusion, the embedding axis captures new data-driven function interactions that are not described in the ontology. These interactions does not represent the semantic similarity between the terms, but their function interaction that give rise for high-order cellular processes. And our next question was to investigate in detail which are the high-order cellular processes that each axis represents. However, here we find a problem, and is that manually validating these processes is really hard, because on average each axis have 11 GO terms associated. Thus there is a lot number to manually do it, and in order to overcome this problem, we propose to summarize the set of GO terms associated with an axis into a high-level functional annotation that we term axis-specific functional annotation, or ASFA for short. An ASFA is not more than a set of keywords that we extract from the descriptions of the GO terms that are associated with a given axis and summarize all of them. In order to get these keywords, we adapt the TF-IDF that are usually applied in NLP to our problem. In general, or briefly, this TF-IDF is a statistic that reports how important a word is to a document, for instance, a chapter, in a corpus, for instance, a textbook. In this case, we're going to adapt this TF-IDF by considering as a corpus the set of GO terms that are associated with our axis, and as a document each of the axis itself. By doing that, we can apply this formula that we can discuss if you want after the talk, and get the keywords for each of the axes. After getting these ASFAs, we assess first of all if they correctly summarizes the GO terms, and secondly, if they describe coherent biological functions by literature curation. Here I'm showing a few examples, in this case axis 79, axis 120, and as you can see, if you go for the ASFAs of 79, let's say, we see that the ASFA indeed describe a coherent function that is related with the development of different organs. In conclusion, the embedding embedding access captures coherent, complex functions and opens a new opportunity for the development of data-driven ontologies using the ASFAs. Not only that, but the ASFAs can also be used to summarize the functional organization of the cell from a new perspective and with new functions that arises from the data. To conclude this presentation, I would like to show one of the applications of our ASFAs in biology. In our project, we decided to apply the ASFAs to gain insights into the evolutionary story of humans. The evolutionary story of humans has been always investigated from the point of view of phylogenetics, meaning that it's usually assessed by comparing the similarity of different sequences between different species. However, with our new methodology, we believe that we can use a data-driven approach to get information from omic data, and with that, finding new connections that arises from the data between different species. And this is quite important because understanding the evolution could be the key to understand different diseases, to also understand how molecular mechanisms work across different species, and also to get more information about our story. In order to apply our ASFAs in this context, we propose the following method. We propose for it to divide its ASFA based on its conservation degree. The conservation degree of an ASFA is something that we introduce also in this work, and basically it corresponds to the union of the different taxons in which the GO terms associated with its corresponding axis appeared. But basically, just forget about that. Basically, the conservation degree is a measure that is going to tell you how much of the function represented by the ASFA is conserved. The higher the conservation degree, the more conserved the function is. We also propose to divide its ASFA based on the taxons according to which type of taxons it is associated with. And by doing that, we can divide the ASFA in prokaryote if all the taxons, at least one taxon, is a prokaryote, in vertebrates if all the taxons are vertebrates, or in eukaryotes if at least one of the taxons is eukaryote, but none of them is a prokaryote. By doing that, we divide the ASFAs, and interestingly, the first thing that we find is that there exist evolutionary patterns, meaning that the functions described by ASFAs that arises from prokaryotes are the ones that are more conserved through evolution, and the other way around, the new ones are those that appear in vertebrates. By comparing the ASFAs in this context, from evolutionary perspective, we also find new connection between different species. For instance, we demonstrate that the neural synapses in vertebrates arises from the molecular mechanisms that existed already in prokaryotes. We also validate that, as is known, the adaptive immune system arises in vertebrates for the first time. In conclusion, here we introduce a method that shifts the exploration of the network embedding space from the organization of the embedded entities to the space itself. We find that the embedding axis captures new interaction between GO terms that are not included in the ontology but are yet biological coherent. We also show that the embedding axis represents high-order biological processes that we term ASFAs, and we apply our ASFAs to get insights into the biological history of humans. To conclude, I just want to mention here that we know that our method has a limitation because not all the axes have a GO term associated. Thus, we didn't find an ASFA, or we didn't find the high-order cellular processes represented by 30% of axes that does not have GO terms. We go over this limitation in the paper by also associating genes to the axis of the embedding space and demonstrating that using these genes instead of the GO terms can be also useful in order to get the ASFA that we validate, and we show that it's biological coherent. Not only that, but also complements the information that we can get from the GO terms. Finally, just say thank you to all here, and just remind to you that if you are interested in other projects that the group are going to present, you can go, you can attend to the poster session that will be held on Monday. Thank you all, and see you around.